Hey!
Hey!
Hey, Platypus!
Welcome to Platinum, where future top notches are made. I'm Cantarara.
And I'm Kay Gonzalez. Today we'll explore the fluid mosaic model.
Come on and join us in our journey to become top 1.
Where's Kent? Anyways, before we talk about the fluid mosaic model, we first must review the key concepts to understand the model. Oh look, there's Kent!
Question number 1. What is the selectively permeable barrier that surrounds a cell, separating its contents from the external environment? Correct. It's the cell membrane. Oh, hi Kate.
Question number 2. What is the primary component of the cell membrane? That's right! Phospholipids! Back to Kent!
Oh, that made my head hurt quite a bit. Let's not do that again. Question number 3. True or false? Phospholipids have a polar head and a non-polar tail. Correct. Phospholipids do have a polar head and a non-polar tail. Back to you, Kate.
Question number 4. True or false? The outer part of the phospholipid bilayer is hydrophobic, while the inner part is hydrophilic. Correct. It's false. The inner part is hydrophobic, while the outer part is hydrophilic.
Oops, sorry, Kate. So it seems like you're all ready to go. Now let's talk about the fluid mosaic model.
By the end of the lesson, students should be able to, first, define biological membranes and explain their importance in cells. Second, describe the fluid mosaic model, including the structure and roles of each component. Third, illustrate how membrane fluidity is maintained. And fourth, identify how different substances move across membranes.
Hey, it's me, Kate again. I'll serve as an introduction. Have you ever wondered how cells can stay alive, communicate, and maintain their internal balance in such a busy and unpredictable environment? The answer lies in the biological membrane. A biological membrane, also known as the cell membrane, is a thin, flexible layer that forms the outermost layer of the cell. It separates the internal environment of the cell from the external environment. It acts like a protective barrier that controls what enters and exits the cell. But it does more than just protect. First, the membrane helps the cell maintain its shape and structure. While it's flexible, it still gives the cell a certain form while staying together. Second, it controls the movement of substances in and out of the cell. This includes oxygen, nutrients like glucose, and waste products like carbon dioxide or urea. This process is essential for the cell's survival and function. Third, the biological membrane is involved in cell communication. It contains proteins that can receive chemical signals like hormones and send messages into the cell so it knows how to respond. Fourth, it helps cell recognition and interaction. Cells have markers on their membranes that allow them to recognize each other and work together, especially important in forming tissues and organs. Finally, the membrane plays a role in transport mechanisms like diffusion, osmosis, active transport, and endocytosis, processes you'll study in detail later on. So, in summary, the biological membrane is not just a boundary, it's an active, essential part of the cell, responsible for protecting, communicating, and regulating everything the cell needs to live and function properly. So now we're done discussing the biological membrane. To test your knowledge retention of the discussion, I will ask a few questions. True or false? Question number 1. The membrane only functions as a protective barrier that separates the cells from its surroundings. The answer is false. It does more than just protect. It also communicates, transports, and helps with recognition. Question number 2. Proteins in the cell membrane help the cell receive signals such as hormones from the environment. The answer is true. Membrane proteins receive chemical signals like hormones. Question number 3. Flexibility of the biological membrane prevents it from playing any structural role in maintaining the cell's form. The answer is false. Flexibility doesn't prevent structure. It actually helps the membrane maintain its shape. Wow, great job answering the questions. Now, that's all the questions I have, and thank you for listening to my part of the discussion.
Hey, it's me, Gab, and let's talk about the fluid mosaic model.
To start off, let's explain its origins. The fluid mosaic model was first proposed by S.J. Singer and Garth L. Nicholson in 1972 to explain the structure of the plasma membrane. But it has some history before we get there. It all started back in 1895 when Ernest Overton hypothesized that cell membranes were made out of lipids. This idea stemmed from the observation that lipid-soluble substances readily diffuse across cell membranes. Fast forward to 1925, where Evert Gorter and Francois Grandel found that red blood cell membranes are formed by a fatty layer 2 molecules thick, which describes the bilipid nature of the cell membrane. Even later, in 1935, Hugh Davison and James Danieli proposed that lipid membranes are layers composed by proteins and lipids with pore-like structures that allow specific permeability for certain molecules. All these and more contributed to the general idea we have of cell membranes today, the fluid mosaic model. This model explains the structure of the plasma membrane of animal cells as a mosaic of components such as phospholipids, proteins, cholesterols, and carbohydrates, which all contribute to its semi-permeability, fluidity, and mosaic attribute. First, let's talk about semi-permeability. Membrane semi-permeability is a consequence of their inner hydrophobic environment generated by the lipid fatty acid chains. This hydrophobic space is difficult to be crossed by molecules having net electric charge. In other words, polar or charged substances such as sodium ions, potassium ions, chloride ions, and amino acids have a difficult time freely passing through the cell membrane compared to small molecules without electric charges such as CO2, N2, and O2, and molecules with high solubility in fat such as ethanol, which can cross membranes almost freely by passive diffusion. Second, the membrane's fluidity. Fluidity refers to the ability of molecules to move within membranes, the lipid bilayer, made of phospholipids. It's not rigid. Its components have constant lateral movement within their own leaflet. This dynamic behavior is primarily influenced by three key factors, those being the degree of fatty acid saturation, because unsaturated fatty acids increase fluidity, temperature, because higher temperatures increase molecular motion, and cholesterol content, because cholesterol modulates fluidity by preventing excessive rigidity at low temps and excessive fluidity at high temps. The fluid nature of membranes is essential for numerous cellular functions, including membrane transportation and cell division. Now, why is it called fluid mosaic model? It's because a diverse array of proteins, carbohydrates, and cholesterol, which are embedded within or attached to the lipid bilayer, are arranged in a way that resembles a mosaic. But don't let it resembling a mosaic fool you into thinking the arrangement is random. It's not. The distribution is organized to facilitate specific cellular functions.
By studying the fluid mosaic model, we can gain insight into how life operates at a cellular level and how disruptions in membrane structure can lead to disease, making this very essential for both basic science and medical research.
I am now going to ask a few questions to test for your knowledge retention. Number 1. Who first proposed the fluid mosaic model? A. Hugh Davison and James Danieli. B. Evert Gorter and Francois Grandel. C. S.J. Singer and Garth Nicholson. Or D. Ernest Overton and S.J. Singer. The answer for this question is letter C, S.J. Singer and Garth Nicholson. Number 2. Which part of the membrane is responsible for its semi-permeability? Is it A, cholesterol, B, carbohydrate chains, C, protein channels, or D, the hydrophobic core? The answer for this question is letter D, the hydrophobic core. Number 3. Which group of molecules struggles to pass through the membrane without assistance? A, sodium and potassium ions, B, ethanol and O2, C, CO2 and O2, or D, water and ethanol? The correct answer is letter A, sodium and potassium ions. Those are all of the questions asked in this part of the lecture. Good job if you got them right.
Hi, I'm Jovian. Think of the cell as a high-tech laboratory and the membrane, its security system, control panel, and communication hub on all one. Without the membrane, a cell is just a soup of molecules with no organization or control. Phospholipids are known as the structural foundation or the primary building blocks of the plasma membrane. They are amphipathic molecules, which means they have a hydrophilic or water-loving head and hydrophobic or water-fearing tails. These molecules arrange themselves into a bilayer with the tails pointing inward, shielded from water, and the heads facing outward towards the watery environments inside and outside the cell. This bilayer acts as a semi-permeable barrier between the inside and outside of the cell, allowing small, non-polar molecules like oxygen and carbon dioxide to pass through, while restricting large or charged molecules. It also allows lateral movement of membrane components and acts as a base for embedding proteins and cholesterol. Next up is cholesterol. It is a lipid molecule that fits snugly between phospholipids in the membrane. While it might get a bad reputation in health, cholesterol is vital for membrane function. It stabilizes the membrane by preventing it from becoming too fluid at high temperatures and too rigid at low temperatures. It also reduces permeability to small water-soluble molecules, which means it limits the ability of certain molecules to pass through the phospholipid bilayer, thus making the membrane more selective. It also helps form lipid rafts, which are microdomains rich in cholesterol and proteins that serve as platforms for cell signaling and transport. Now that we've talked about the phospholipid bilayer and cholesterol, let's move on to membrane proteins. They are responsible for most of the membrane-specific functions. They are like specialized workers embedded within or attached to the membrane, each with a specific job to help the cell interact with its environment, communicate, and transport substances. In the fluid Mosaic model, proteins are said to float in a sea of lipids, hence the term mosaic. Let's break these proteins down into two major types. First is the integral proteins or intrinsic proteins. These are embedded within the phospholipid bilayer and often spans across it. Their functions include transport of molecules, signal reception, cell recognition, and enzymatic activity. The second major type of protein is the peripheral or extrinsic proteins. These are proteins that are not embedded in the lipid bilayer. Instead, they are loosely attached to either the outer or inner surface of the membrane, often anchored to integral proteins or to phospholipid heads. They do not penetrate the hydrophobic core. It acts as enzymes or signaling molecules, provides structural support by anchoring to the cytoskeleton of the cell, and assists in cell communication and recognition. Lastly, we have carbohydrates. While they may not be as abundant as lipids or proteins, carbohydrates play a critical role in how cells communicate, identify each other, and protect themselves. Carbohydrates are not embedded in the bilayer, but are attached to proteins forming glycoproteins or lipids forming glycolipids on the extracellular surface of the membrane. Together, these sugar-coated molecules form the glycocalyx, a fuzzy, protective layer surrounding the cell membrane. Now, let's take a quick quiz to check your understanding of what we've covered. The three major components of the plasma membrane are cholesterol, phospholipids, and proteins. True or false? True. The three core components are phospholipids, cholesterol, and proteins. The amphipathic nature of phospholipids allows them to form a bilayer with hydrophilic heads facing inward. True or false? False. The hydrophilic heads face outwards. Carbohydrates may not be embedded within the lipid bilayer, but it is attached to the cytoplasmic side of the membrane to form glycoproteins and glycolipids. Is this true or false? False. They are attached to the extracellular surface, not the cytoplasmic side. Great job on completing the quiz. Hopefully, it helped solidify the main ideas we covered.
Hey, it's me again, Ken. So now we're down to our very last part of our discussion, which is the membrane asymmetry of the fluid Mosaic model. So what does membrane asymmetry mean? This means that the two layers are structurally, physically, and functionally different. This stems from an uneven distribution of molecules between the two lipid layers, also known as your leaflets.
We can tell that the inner and outer leaflets of a cell membrane is asymmetrical because of three key differences. 1. The difference in composition of lipids, proteins, and carbohydrates. 2. The difference in the orientation and positioning of the proteins. And 3. The difference in the enzymatic activities of the two sides of the membrane. As mentioned by Jobian, molecules such as lipids, proteins, and carbohydrates attach to both the outer and inner leaflets of the cell membrane. But what exactly is the difference between the molecules in the two hemilayers? The majority of lipids in the outer hemilayer of the plasma membrane bear choline, such as phosphatidylcholine and sphingomyelin, whereas the inner hemilayer contains more phosphatidylethanolamine, phosphatidylinositol, and phosphatidylserine. The presence of some lipids in a particular hemilayer is important for cell functions. For example, phosphatidylinositol is mostly located in the inner hemilayer of the plasma membrane and can be split into two molecules by some phospholipases. One of the molecule travels through the cytosol as a second messenger. Why is asymmetry important? The asymmetry determines an unequal electric charge distribution at both sides of the membrane, which helps to generate the membrane potential. It also allows the association of specific proteins with a particular surface of the membrane due to the distinct environment created by the electrochemical properties of lipid heads, which is different between both sides. The membrane symmetry also provides physical properties to membranes. For example, in the plasma membrane, it is thought that the formation of vesicles is easier towards the cytosol, i.e. membrane invasions are forwarded toward the cytosol. Why is it important to analyze the specific asymmetrical molecules of the cell membrane? Disorganization of the membrane asymmetry is sometimes a signal indicating that something is going wrong in the cell. For example, during apoptosis or regulated cell death, the outer hemilayer of the plasma membrane exhibits lipids that are typical of the inner hemilayer. These lipids are recognized by macrophages that finally remove the cell. In fact, there are proteins in the plasma membrane of healthy cells surveying for phosphatidylserine in the outer hemilayer. When they find one, it is quickly returned into the inner hemilayer. The loss of asymmetry is also important for blood coagulation. Some viruses having a membrane envelope show phosphatidylserine in the outer hemilayer so that they can be incorporated more easily by phagocytosis or macropinocytosis. Okay, so now it's time for the huge, our quiz part/segment. Question number 1. Which of the following is not a contributing factor to membrane asymmetry? Is it A. Difference in lipid composition, B. Identical protein orientation, C. Varying enzymatic activity, or D. Different protein positioning? Correct. It's B. Identical protein orientation. Question number 2. What happens to phosphatidylinositol after being cleaved? A. It is excreted from the cell. B. It becomes embedded in both leaflets. C. One of its products acts as a second messenger. Or D. It produces ATP. Correct! It is C. One of its products acts as a second messenger. And your third and last question, which lipid, when exposed on the outer membrane during apoptosis, signals macrophages? Is it A. Phosphatidylinositol, B. Your sphingomyelin, C. Phosphatidylserine, or D. Glucose? Correct! The answer is C. Your phosphatidylserine. So that's it for membrane asymmetry, and hopefully you've learned a lot from this lesson.
Remember, strive, excel, succeed.
This is Platinum, where future top-notchers are made. See you on the next review.